So today's lectures are going to be about cell structure. So we're going to begin by reviewing the tenets of the cell theory. And those tenets state that all organisms are made up of at least one cell, that cells are the fundamental unit of life, and that all cells come from other cells. And we've been slowly talking about the small monomers that form macromolecules that build our cell. So we have proteins and membranes and nucleic acids. And we've started to talk a little bit about, or we've talked a lot about how cells can metabolize, the things that cells can eat. And now we're going to start to take a look at organelles and really some more complex interactions that take place within the cell. But before we really dive into that, I want to spend a little bit of time talking about the different kinds of cells. So based on morphology or form, cells were originally divided into two fundamental types. Those being eukaryotes that have a membrane-bound nucleus and prokaryotes that lack a membrane-bound nucleus. However, based on phylogeny or evolutionary history, right, how far back our common ancestor goes, we actually know that organisms are divided into three domains. We have eukaryotes belonging in this eukarya domain, but prokaryotes actually belong to these two separate domains of life. And those are bacteria, which diverged from us a pretty long time ago, and archaea that are more evolutionarily closely related to us. So we're going to focus a lot today in this first video on bacteria and what elements are present in bacteria. And then the next lecture will focus on eukarya. But all of these cells, all basic cells have these fundamental units. So all cells have proteins, and these form most of the cell's basic functions. All cells have nucleic acids that store and transmit and process information. All cells have carbohydrates that they can use for chemical energy, carbon, support, and also identity. And all cells have a plasma membrane that serves as a selectively permeable membrane to separate the insides of the cell from the outside environment. We've talked about how all of these structures are built and how they contribute to some functions in the cell. So these are shared between all three domains of life. And like I said, we're going to focus on bacteria and archaea and really hone in on bacteria and how these cells are different from eukarya and what are the basic building blocks of a bacterial cell. So bacteria and archaeal cells possess DNA. So all cells have DNA, but it's in a region called the nucleoid. And so down here is an image of a bacterial cell using transmission electron microscopy. And the way that this technique works is that they can stain the cells with some sort of chemical that will, when fired with a beam of electrons, it will turn dark if there's some sort of organic structure there. And so where it's dark, you can see these are structures within the cell. And so the nucleoid is usually one single chromosome and it's circular. And this is true for most bacteria, that they have this one chromosome that is circular. And you can even see here's another bacterial this one's round. And the nucleolus is shown here in the center of the cell. And so this is the area in which the DNA resides, but it's not membrane bound like a nucleus. Bacteria can also have plasmids, and so these are extra chromosomal pieces of DNA, and they can carry important genes for a variety of things, including antibiotic resistance. But this is separate from the single chromosome that defines that bacteria. So bacterial cells also have ribosomes. Ribosomes are complexes of protein and rRNA. And so here again is that same picture of that round bacteria. And the ribosomes are depicted by these dark structures kind of on the perimeter of the cell. And what the rRNA does is it directs the catalytic step of protein synthesis. So recall from the central dogma that we have DNA that gets converted into RNA, and then the RNA gets converted into protein, right? So we have to link together those amino acids. And the rRNA in ribosomes is what directs the catalytic step of protein synthesis. And so sometimes rRNA is also called a ribozyme or catalytic rRNA to make reference to it directing those catalytic steps. And kind of an interesting fact is that eukaryotic and prokaryotic ribosomes are different as a result of divergent evolution. And so a lot of antibiotics on the market target ribosomes from bacterial species, but those same antibiotics don't affect our ribosomes because they're just different enough to give specificity to those drugs. Many bacteria also have different structures that grow from their plasma membrane that enable some sort of movement or attachment. And so a common type of structure you might see is a flagella. These are long filaments that rotate or propel the cell, and so I'm sure you can imagine what this looks like. But a lot of bacteria also produce these structures called fimbriae. These are needle-like projections that promote attachment to other cells or surfaces. So here's an example. This is an E. coli cell. This is actually a pathogenic E. coli cell. And it's producing these fimbriae structures, these needle-like projections. And these particular fimbriae are really important for the colonization of these pathogenic E. coli. They're going to cause the symptoms of food poisoning. And so this helps them attach to the surfaces in our gut and helps them reside there and kind of take over. And so maybe targeting these structures, these fimbriae, could be a therapy to kind of relieve the symptoms of food poisoning. Okay. Some newer findings also suggest that some bacterial cells have limited cytoskeleton and primitive organelles. And I do want to stress that these are only some types of bacteria, right? So most bacteria don't really have organelles, and the ones that do, they're very primitive structures. So the limited cytoskeleton, what it does is it helps the cell divide, and we'll talk more about cell division later in the quarter. And some bacteria also have primitive organelles. And what these organelles can do is store things like calcium. This is a really important ion. Some primitive organelles hold crystal minerals, and this helps kind of create some sort of magnetic sensing system. And so the bacteria knows where to navigate. And some types of bacteria which are photosynthetic, have internal membranes that support photosynthesis. And so they can convert energy from the sun into chemical energy. And it doesn't happen in the chloroplasts like we talked about in plants. Instead, it happens in these folds in the plasma membrane. And so here's an example of a cyanobacteria. This is the same kind of bacteria that causes harmful algal blooms. And here's a transmission electron microscopy image from a paper in EMBO. And what you're seeing here are these folds coming off from the plasma membrane. And what these folds are primitive thylakoid structures that contain the enzymes necessary and the proteins necessary to convert light energy into chemical energy. And that can be used to fix carbon dioxide. Some other differences between bacteria and archaea is that they both have a plasma membrane, but that plasma membrane differs in structure. So recall that the plasma membrane is made up of phospholipids, and phospholipids have usually a glycerol molecule and a phosphate, and they're attached by ester linkages to fatty acid tails. Well, this is the case for eukaryotic and bacterial cells, but these archaeal cells, instead of having fatty acid tails, they have isoprenoids in their tails. So this distinguishes archaeal cells evolutionarily from bacteria. They also differ in their cell wall components. So moving out from the plasma membrane, bacteria need a cell wall. And the cell wall is very important because these are single cell organisms. And so they're interacting with their environment. And so if they enter into an environment with a hypotonic or a hypertonic solution, they need a way to kind of protect the cell from losing all their water or kind of gaining a flood of water into the cell and causing that cell to burst. And so the cell wall helps with this function. So I'm going to focus on the components of a bacterial cell wall. So on top of their lipid plasma membrane, there's two kinds of cell walls. And so in gram-positive bacteria, there's a thick layer of peptidoglycan. And peptidoglycan, as we talked about, is this carbohydrate that's formed by these alternating monomers, NAG-NAM, NAG-NAM, and then they're linked together in layers via peptide bonds. And so this forms a really tough, thick cell wall. And we can see if we have a thick cell wall by doing this technique called gram staining. And if you ever take a microbiology class here at Cal Poly, you'll be able to do that technique. We also have gram-negative bacteria. And in this case, that peptidoglycan layer is a little bit shorter, not as thick. And then on the very most outer membrane has this structure called lipopolysaccharide. So lipo because it's part lipid, and then also sugar for polysaccharide. So this just distinguishes different kinds of bacteria based on the shape of their cell wall. And bacteria come in a wide variety of shapes and sizes. So here are some of the different shapes that a bacteria can come in and our name for them. So usually we call this very round shape of bacteria, we call this a cocci. And as it begins to stretch out into a bacillus, which is this pill-shaped structure, we can kind of see this one here as a coccobacillus. We can see there's also bacteria that form, we call this a club shape or vibrio. Then there's ones that spiral around like the spirillum, or this one here, the spirochete. And so these are very interesting shapes that these bacteria take on. Bacteria can also vary dramatically in size, and so we can get a bacteria smaller than 0.2 microns in diameter. And then just last year, we discovered a new largest bacteria, this T. magnifica, and it's approximately 2 to 4 centimeters. And so what you're looking at here is a dime. And this is not a microorganism because we can see it because it's quite large. And so we'll talk in the next video about really most bacteria are limited in size by their inability to take on more organelles or more complex organelles that can specialize that cell in a variety of ways. So to summarize what I said so far, bacteria and archaea don't have a nucleus. They do have DNA. The bacteria and archaea have slightly different phospholipids. They have a different cell membrane. So bacteria have cell wall. Bacteria have peptidoglycan in their cell wall. They're both small, right? They're quite small, quite a bit smaller than these eukarya, even though large examples of bacteria exist. They all contain ribosomes, and in general, bacteria and archaea have one chromosome. So that's it for this video, and I'll see you in the next one. Thanks, bye.